Hello, everyone. Welcome to the lectures for General Biology 2. My name is Kyle Rose. I'll be the instructor for this course. We're going to start with Chapter 27, which deals specifically with protists. So when we look at protists in this chapter, we're going to look at what a protist is. We're going to describe its role in evolutionary trees, its importance in medicine and ecological significance. We'll talk about how protists paved the way for all multicellular organisms through the endosymbiont theory. We'll talk about how these things eat, how they move, and how they reproduce as a whole, right? So here are some examples of protists: slime molds, amoebas, euglenas, dinoflagellates, paramecium, diatoms, microalgae. So these all fall under the umbrella of protists. And so in this chapter, like I said, we're going to be looking at various things that will describe the background of protists. So throughout this whole course, we're going to be looking at different organisms that fall under the domain Eukarya. So there's three domains of life. We've got the Archaea, which are sort of these extreme organisms, these extreme bacteria-type organisms that live in areas where other things don't live. We've got our bacteria, and then we've got our Eukarya. Right. So what makes these things different is they've got essentially a nucleus. All right. They've got a nucleus. They've got organelles. And eukarya can be single-celled, so we can see single-celled organisms like protists in some cases, where they are eukaryotic, they have a nucleus, but they are single-celled. And we can see multicellular organisms as big as trees and blue whales also falling under the same domain. Okay? So obviously this is a very diverse group. Okay? There's fundamental features, though, that distinguish organisms in domain Eukarya from bacteria and Archaea, the other two domains of life. So most of these are going to be larger than bacteria, but not always. They have organelles. That's a distinguishing factor. So they're going to have things like Golgi complex and endoplasmic reticulum, and all of those specialized cellular organelles that have their own membrane around them. All right. They also have a more extensive cytoskeleton, so in their cells they've got more structure to hold everything in place. All right, the synaptomorphic characteristic of all organisms in domain Eukarya, that means something that's shared by all of them, is a nuclear envelope. All right, so it really defines this. Multicellularity involved multiple times in eukaryotes, so we see different types of multicellular organisms in this domain. And we see that these can reproduce either asexually via mitosis, or sexually via meiosis, in the process of egg and sperm fertilization. So let's specifically look at protists. So protists are all the eukaryotes that are not fungi, they're not green plants, and they're not animals. So we'll talk about that distinction between green plants and the plants that we're going to see that fall under this category. All right. Protists tend to live in environments that are just near water. So wet soils, aquatic habitats, or even inside the bodies of other organisms, right? So we're going to see a lot of parasites falling under this group of protists. These organisms are paraphyletic, so that means that protists are going to represent some, but not all of the descendants of a common ancestor. So when we look at the evolutionary tree of a protist, we're going to see that there's branching patterns for these protists that diverge away from where we see other organisms. So in a monophyletic group, all of the descendants would share the common ancestor, right? That's not the case in protists. So that's why we see a whole lot of diversity here. And these are, again, being extremely diverse organisms. All right. So here's, a look at some of these different types of places where we find protists and what they look like. So in the open ocean, we find microscopic protists such as these diatoms, right? So took a scoop of ocean water and you were looking under the microscope, you'd see tens of thousands of these little diatoms in there, and these are all examples of protists. They have a nucleus. They're single-celled, but they are eukaryotic. Shallow coastal water. Here's an example of an underwater kelp forest. This is a large multicellular protist. So gigantic protist. So kelp is a protist. It's different than green plants, and these form underwater forests. All right. Protists such as the brown algae, particularly abundant in tidal habitats. So we see these here, these intertidal habitats containing these protists. Notice again that we're looking at places that are in and around water where we find these protists commonly. So why should you care about protists? right? Other than the fact that you'll see them on your first exam. But a biologist, and those of you that are going to go on to study further in biology, whatever facet, it's important to look at all aspects, right? And protists represent a really specific niche here. So there's definitely impacts on human health and welfare. So in terms of um, public health, we're going to look specifically at things like the Irish potato famine, malaria, which are diseases that are caused. Malaria is a disease caused by a protist. So there's a huge impact here on medicine. Ecological importance, right? So we're going to see that these can either disrupt an ecosystem or they're going to be fundamental to an ecosystem, right? And by understanding protists, we can understand the evolution of other groups, plants, fungi, and animals, which we'll be studying later on in the course. So one of the best examples we can talk about as the impact of human health is malaria. So this parasitic protist called Plasmodium is transferred to humans from mosquitoes, and this causes malaria. So it's really the world's most chronic health problem. 200 million people worldwide are affected by this malaria, and it kills about 600,000 a year. Okay, so specifically, there's 5 species of this protist genus Plasmodium. Okay, so basically, what happens here is we're gonna take a look at this, but the Plasmodium cells are going to enter the bloodstream through a mosquito bite, and it's going to infect liver cells, ultimately infecting your red blood cells and destroying them. Okay, so this is obviously something that's going to be really detrimental to a person that is infected with malaria. All right, so here's a look at the cycle here between the Plasmodium parasite and its human host. Right? So if we take a look here, Plasmodium type cell present in mosquito saliva infects the humans through the bite. Okay, so Plasmodium gets into the cells here, and you can see that when it gets into those cells, it's going to destroy that cell ultimately. So this would be infecting the liver cells here, and as these cells continue to divide, they're going to continue to produce more infected cells. All right. Once the Plasmodium goes out and it infects the red blood cells, this is where it really becomes a major problem. Okay, so Plasmodium cell types present in human blood that infect mosquitoes. So then the cycle continues here, and you can see that through a mosquito bite, the male and female gametes can end up going back into the mosquito, and the cycle can continue between humans and between the mosquitoes, right? So if the mosquito bites a person with infected blood, or a mosquito that has that infected blood gives it back to a different person, we see sort of this alternation between the two, right? So this is kind of a basic look at how the Plasmodium is transmitted via mosquito to a human host. So we've spent a good amount of time studying malaria. It's been around for a long time, but there's not a whole lot in terms of effective and sustainable measures to control it. So drugs that are used to control, this disease the disease has evolved in response to that resistance, and researchers have been unable to develop a really effective vaccine because of that evolution. All right. For the mosquitoes, right? If we try to control the mosquito population with insecticides, so let's say, okay, we wipe out all the mosquitoes, and if we wipe out all the mosquitoes, essentially we'd be able to wipe out malaria. The problem is that the mosquitoes have responded to that by evolving resistance to insecticides. So the mosquitoes that are resistant to the insecticide through natural selection will be the ones to reproduce, and then we'd be left with only the mosquitoes that are resistant. So that's an example of how a protist that one protist that causes human disease. If you take a look in your textbook and think it's chapter or table 27.1, there's a list of a lot of other protists that cause disease that you can take a look at. And I think in your mastering bio, you'll see some of those in your assignment. In terms of ecological influences, so one of the things that protists can do is they can cause these algal blooms. So a bloom is going to occur when a protist population grows rapidly, reaches high densities in aquatic environments. So specifically, this is caused by protists called dinoflagellates. All right, and these um, dinoflagellates are going to lead to harmful algal blooms. So this is harmful to humans because toxins build up in clams and other shellfish. So people who eat those shellfish can then get sick or die. All right. So if we have a lot of these toxin-producing dinoflagellates, it's obviously going to make its way into the ecosystem. And when we eat those poisoned shellfish, it causes a problem. So here's a look at those dinoflagellates. Okay, they kind of have a brown color. You can see the bloom here. They're photosynthetic. As they bloom, they produce those toxins, and it causes an increase in this xanthophyll, which is a pigment that's going to discolor the water. And like I said, this is going to produce these toxins that are produced by the dinoflagellates and get into the shellfish and other organisms that we rely on for food, and not only affects the ecosystem, but also affects humans and fishing and things like that. Right? So kind of getting into that same idea, protists obviously play a key role in aquatic food chains. So even though they represent just 10% of named eukaryotic species, they are very abundant. Okay, so these generally fall into the role of primary producers. So that means that they're going to produce chemical energy by photosynthesis. So they're going to be at really the bottom, I guess you could say, of the food chain or the food pyramid. So if we lose those primary producers, everything up the chain is going to be lost. Production by marine protists, so that means that how much of what they take in sunlight is turned into glucose, represents half the total carbon dioxide that's fixed on Earth. Okay, so carbon dioxide in the atmosphere builds up, it's toxic. So these marine protists take in that carbon dioxide, almost half of it, and turn it into aquatic oxygen that's then used by fish. So really, really important that we have these protists that are doing this photosynthesis and primary producers. Okay. Plankton, diatoms, and other small organisms that drift in open oceans or lakes are going to be critical in the aquatic food chain. Another example of protists that are very, very important. I mentioned the food chain as being a really important part here. So nutritional relationships among organisms, the flow of energy is shown in a food chain, and the species at the base of the food chain in aquatic environments are protists. So where plants on land provide the basis for the bottom of the food chain for terrestrial ecosystems or land ecosystems, these protists provide it in the water. So protists could help to limit global climate change and potentially take up large quantities of carbon dioxide, and they carry it to the bottom of the ocean, where they die. So phytoplankton, for example, do this. So the global carbon cycle is the movement of carbon atoms from carbon dioxide in the atmosphere to organisms on land or the ocean, then back to the atmosphere. All right, and generally this happens if you think about it on the land, right? You've got the carbon dioxide in the atmosphere. The plants take in that CO2. They go through the process of photosynthesis and they turn the CO2 into glucose. So these protists do the same thing. So to decrease CO2 concentrations in the atmosphere, we could increase the amount of carbon stored in aquatic and terrestrial environments. So protists act as carbon sinks. All right, so they're long-lived carbon repository. And by doing this, we can see that sedimentary rocks from protists with shells made of calcium carbonate, petroleum, accumulation of dead bacteria, archaea, and protists at the bottom of the ocean. So like I said, these things will take in the carbon dioxide, and then they'll ultimately go to the bottom of the ocean and die, and that's where the carbon will live. So long-lived carbon sinks here, which is why these protists are very important in limiting the amount of CO2 in the atmosphere and ultimately global climate change. Here's a look at kind of what I described here. So carbon dioxide goes into the aquatic environment, the primary producers. All right. These primary producers will be eaten by primary consumers, right? ultimately, they'll and the carbon will go from the primary producers to the primary consumers. They'll decompose and they'll be, taken out by scavengers, and the carbon will continue to sink here until it gets to the bottom of the ocean. All right. Um, that's one pathway that the carbon can take. The primary producers could die themselves and sink. Okay. Which is shown here and here. Okay. And here, the primary consumers could sink. Either way, we see a situation where no matter how the carbon flows, or cycles, I should say, it makes its way accumulating to the bottom of the ocean. Okay, so what are some of the ways that biologists study protists? So they study them through microscopy or cell structures, studying cell structures. So we can look and see that there's, many protists have characteristic overall forms. They have organelles with distinctive features. We can see that. For example, there's flagella with hair-like projections that we see on these protists. And specifically the ones that were on the protists that were in the potato famine, could be linked to that problem. Molecular phylogenies. So molecular phylogenies, here what we're looking at is DNA sequences, DNA sequence analyses that demonstrate that the 7 major eukaryotic groups are indeed going to be monophyletic. So even though protists are considered a paraphyletic group, we can see that eukaryotes do have distinct monophyletic branches. You can also look at direct sequencing and look at the DNA directly of the protists. Okay, so here we're looking and comparing DNA of the protists from different phylogenetic groups, and here we're looking at protists directly. So it's difficult to find overall patterns in the links of in the evolution of protists. All right, so there's different themes that we see that we'll talk about here. But about 1 billion years ago, the eukaryotic lineages split, and there's been a great deal of divergence among protists. Okay, so there's no synaptomorphies. Okay, so there's no shared derived traits. All right, among the protists, which is why we say that they are paraphyletic. All right. Data on the morphology and phylogenetic analyses of DNA sequence indicate that there is a whole lot that we can look at here in protist diversity, all right, which we'll see. And we'll look back and trace some of that diversity. So if we look specifically at the microscopy, like I said, this is one way of studying. We see that we can group them by their cell structure and distinctive organelles. So by looking under the light microscope, we get an idea of what these things look like more superficially. Electron microscopy allows us to look at the lineages and really take a look at the DNA and things like that. All right. So, for example, Stramenopiles have unusual flagella covered with hollow hair-like structures. That would be something that would be seen as a distinguishing group of that protist that you wouldn't see under the light microscope, but you'd see under the electron microscope. Right. 7 major groups of eukaryotes have been identified on the basis of diagnostic morphological characteristics. Right. So all of this we can come with that understanding. Okay. Here are the distinguishing features or the synaptomorphies of the major lineages of eukaryotes. All right, and you can take a look at these. All right, so we got the amoebozoa, cells lacking cell walls. Okay. Pithecanthta, reproductive cells with a single flagellum at their base. So again, these are just ways of distinguishing these eukaryotes. Excavata, feeding groove or prey or organic debris is digested. Okay, most species lack a typical mitochondria. Plantae, which we're pretty familiar with, cells have chloroplasts with a double membrane. Rhizaria, lacking a cell wall. Alveolata, cells have sac-like structures called alveoli. We'll see some of these later on. Stramenopilia, if flagella are present, cells usually have 2. Right, so these are just ways to distinguish the different types of eukaryotes. Okay, so the second way in which we study protists is by, like I said, evaluating the molecular phylogenies. So morphological data and DNA sequence suggest that the 7 groups are in fact monophyletic, A phylogenetic tree of the 7 lineages shows that the Epithecanta and the Mesozoa form the monophyletic group Uniconta, or one-tail. The other 5 major lineages just form a two-tailed or a biconta. So recognize those two terms, Uniconta and biconta. And that split among these occurred between the uniconts and the biconts. So having the one tail and the two tails. Very interesting. All right, and here's a look at those differences there where we have the uniconta and the biconta. We look at direct sequencing by, again, looking at the specific DNA sequences in a protist or a eukaryote. We can look and see by sampling the soil or the water. We can see DNA sequences of the genes in a sample, use the data to place the organisms on a phylogenetic tree. So again, these are just all ways to study eukaryotes, to study protists specifically. All right, so what are the major themes that occurred in the diversification of protists? So protists are really diverse, like we've seen, okay? So they can be single-celled to giant kelp in the ocean. All right, we can find them in the open ocean. We can find them inside of other organisms. Like we said, morphology and how they look is very different. And looking at the pictures and things like that, you'll get a better idea. All right, we know that protists specifically are paraphyletic, so they don't share a derived characteristic that sets them apart from all other lineages. So what is it that really makes protists protists? We're going to investigate that. Okay. So in protists, what are some of the morphological innovations that evolved? So the earliest eukaryotes we know were probably protist-like. They were single-celled organisms, mitochondria, nucleus, endomembrane system, cytoskeleton, and they did not have a cell wall. They probably swam using a very original, simple flagellum. So how we got to this eukaryotic, so these are early eukaryotes, but how we got to this idea of multicellularity and how we got to this idea of a eukaryotic cell had to do with something called the endosymbiont theory. So this is something you want to understand and recognize. So you know that mitochondria from Bio 1 is the organelle that generates ATP, right? The powerhouse of the cell. So at some point, 2 billion years ago, a bacteria took in another, maybe smaller bacteria, specialized bacteria, and when it took in that bacteria, rather than digesting it or ingesting it, the bacteria actually became a part of its anatomy, and that bacteria that was ingested became the mitochondria. All right. So in a way, it's an example of endosymbiosis. So symbiosis is when individuals of two different species live in physical context. This is an example of endosymbiosis when an organism of one species lives in the cells of another species, right? So this is believed to be the early origin of where the mitochondria came from. Here's a look at that. The host cell, this large cell, ingested the bacterium, and it stayed within the cell, and it did the job of supplying the cell with ATP. So this is generally believed, this endosymbiotic theory, to be how we achieved multicellularity and achieved the eukaryotic cell, I should say. So observation consistent with the endosymbiont theory include the mitochondria is actually similar in size to this alpha proteobacteria, which could have been ingested, right? Mitochondria replicate by fission, which is really interesting because that's how we know bacteria replicate by binary fission. Mitochondria have their own ribosomes. They manufacture their own proteins. They even have their own DNA. They have double membranes consistent with being engulfed, and they have their own genomes. Okay, so like I said, they have their own DNA, circular molecules, similar to bacteria. So it's very likely that at one time the bacteria was, or the mitochondria was, a free swimming bacteria. Okay. The same thing is believed to have occurred with chloroplast in the Plantae. So all photosynthetic protists and plants that we know have chloroplast. So the same idea here, except eukaryotic chloroplasts originated when a protist engulfed something called a cyanobacterium that was photosynthetic. So what kind of evidence do we have that supports this? So chloroplasts have bacteria-like characteristics, kind of like the mitochondria. They have circular DNA. They have something called peptidoglycan in the cell wall, which we see in the cell walls of these cyanobacteria. Okay? So living endosymbiotic cyanobacteria live in the cells of protists and animals. So we see this already being endosymbiotic. So it leads to the possibility that this is how these eukaryotes evolved. All right, so the same sort of idea. We had a predatory protist that ingested this cyanobacterium. The photosynthetic protist is engulfed. Okay, the nucleus is lost from the ingested photosynthetic protist and ultimately became the chloroplast. So in addition to the chloroplast, mitochondria being, essential characteristics here of most of these protists, depending on what they are, the nuclear envelope is something that sets eukaryotes apart, like I said. So the leading hypothesis for where the nuclear envelope or the surrounding to the nucleus came from is it's derived from the inner foldings of the plasma membrane, which is outside of all the cells. So the infoldings would have given rise to the endoplasmic reticulum, which is a folded membrane all around the nucleus. You're picturing that, typical picture of an animal cell. So this nuclear envelope was a good thing because it separated the two processes of transcription and translation. So transcription, remember, takes place inside of the nucleus, right? DNA to RNA, and then translation takes place outside at the ribosomes, which is RNA to protein. So again, showing you again the origin of the nuclear envelope. Okay, so just infoldings of the plasma membrane came in and ultimately surrounded the DNA. So it's really advantageous to have that nucleus protecting the DNA because DNA is a very, fragile kind of molecule, and having that protective covering is really good for it because, it doesn't get degraded as easily. So, all these observations that we've seen, support the infolding hypothesis. We can better control gene translation, gene expression. All right, so this is where I'm going to end part 1 of the chapter 27 lecture.